Hello students, I hope you are enjoying the course. In the last week we learned about the cells and cells types, difference between prokaryotic and eukaryotic cells, and brief function about those cellular components. And we also learned about the plasma membrane, function of plasma membrane in brief, and the function and structure of nucleus, and how the DNA is packaged within the chromosome and chromatin, etc. So in this week we will learn about in details about the next cellular components, which are the endoplasmic reticulum, ribosomes, Golgi apparatus, and mitochondria, etc. in details. So endoplasmic reticulum. So we know that there are two types of endoplasmic reticulum. One is smooth, one is rough. Based on the presence of the ribosomes, they are divided into two types. So this is the nucleus, we know that, and this is the ear, rough ear, and these white hollow tubes are smooth ear. So ear basically extends from the nucleus and extends throughout the cytoplasm and up to plasma membrane. So extensive network of tubules or channels. So these structures are like hollow tubes, tubules, or channels that extends from the nucleus to the plasma membrane. So structure of the ear allows transportation of materials through the internal regions of the cell. So these hollow tubes or tubules allows transportation of the materials through these channels. So when a protein is synthesized in the ribosomes, those protein enters through this ear and travels through this ear, and finally ends up into the Golgi apparatus. We will see that in this week. So smooth ear, we'll see the function of a smooth ear in this slide. In the next slide, we'll be seeing the function of the rough ear. So a smooth ear has many unique enzymes embedded on its surface. So functions are produces produces membrane phospholipids and cellular lipids. So smooth ear basically involves in the transportation or production of the lipids or lipid-based substance. So transfer lipid-based compound. So and the second function is it produces sex hormones, testosterone, estrogen. So sex hormones are basically made up of steroids, and steroids are made up of cholesterol, and cholesterol is one type of lipid. So these three functions involve lipid production or lipid synthesis help. So the smooth ear helps in the transportation or synthesis of the lipid-based compounds. For example, phospholipids, cellular lipids, and steroid hormones, testosterone, and estrogen. So these three functions are quite similar. So the next functions of a smooth ear is detoxification of drugs in the liver. So detoxification means when we ingest a drug and we administer a drug, basically every drug is a toxic substance. So in the liver, the drug gets metabolized or this is called also biotransformation. So every drug converts into less toxic substance in the livers. So liver is mainly involved in drug metabolism or biotransformation, and to some extent kidney and lung is also involved in drug metabolism. So you guys will learn more about drug metabolism and drug biotransformation in the courses like biopharmaceutics and pharmacology, etc. So just know the term detoxification, and detoxification of drugs occur in the liver, and those detoxification or metabolism that involves ER, endoplasmic reticulum, uh, those are termed as microsomal conversion or microsomal metabolism. So ER that involves in the metabolism called microsomal metabolism, etc. And the next function is helps in glucose release into the blood from the liver. So another important function of smooth ER involves liver. So ER helps in the glucose release from the liver into the blood when needed. So the process of glucose synthesis from glucose breakdown from the carbohydrate is called glucogenesis. Basically, glucose are stored as glycogen in the cell, and glucose production or breakdown from glycogen is known as glucogenesis. But gluconeogenesis or neoglucogenesis refers to those glucose production other than the source of carbohydrates. For example, glucose production from proteins or lipids are known as gluconeogenesis, and hepatic refers to liver. So just know the term, you don't have to know much at this stage. Just know the term gluconeogenesis or glucogenesis, etc. So these two function of ER involves liver. The last function involves muscles. So the ER stores calcium ion, and calcium ion helps in the contraction and relaxation of the muscle. So there's three functions. So these three functions are quite similar, that is lipid based functions, and these two functions involve liver, drug detoxification, and gluconeogenesis, that means glucose release, and the last function of smooth ER involves muscle. So going to the next slide, we'll learn about the function of the smooth ER. Sorry, rough ER. So we have learned about the smooth ER in the previous slide. In the next slide, we'll learn about the function of the rough ER. So rough ER, we know that it has ribosomes attached to the exterior of the channels. So these tiny dots are ribosomes. And the rough ERs are located closer to the nuclear membrane. We can see that than the smooth ER. So the function of the rough ER is attached ribosomes are involved in protein synthesis. Yes, we know that ribosomes are involved in protein synthesis, and rough ER is involved in protein development and transport. So those proteins that are synthesized in the ribosomes that travels through this um, channels of the rough ER, and then the rough ER allows these proteins to be transported, and these proteins in the meantime develops into like any modification occurs throughout this transportation. And the last function is that proteins may become parts of the membranes and enzymes or even messengers. So those proteins that goes through this ER, ultimately those end up in the Golgi apparatus, and those protein finally comes out of the apparatus and travels through the cytoplasm, or it can go outside of the cells. So those proteins can become the parts of the cell membrane or cellular organelle membranes, for example, mitochondrial membrane or the nuclear membrane, etc. So membranes are made up of proteins. Or those proteins may become enzymes. So we know that enzymes are proteins, and those enzymes catalyze important biochemical reactions. And messengers. So messengers are also proteins. So messengers are basically like cytokines that communicate between cells to cells. So proteins may become cytokines or messengers. So moving to the next slide. So we know that ribosomes are involved in protein synthesis. So they are unique structure. They do not have external membrane, and they can be found free in the cytoplasm or attached to the surface of the ear. So those ears are called rough ear, and they are made up of a special type of RNA and protein. And those RNA that made the ribosomes are called ribosomal RNA or rRNA. So ribosomes are composed of two subunits, small and large. So this one is large, this one is small. Those two combine to produce ribosome. And those subunits of eukaryotic cells, those two subunits of eukaryotic cells together, they form 80S subunit, and the prokaryotes form 70S subunits. And the ribosomes of the eukaryote cells are larger and denser than those of the prokaryotic cells ribosome. So this figure, in a nutshell, this figure represents the translation process, that means protein synthesis process. So this big guy, the Pac-Man, is the ribosome, and this uh, little guys are transfer RNA. So, and this strand, this nucleotide strand, single strand is mRNA. So RNA are basically single strand nucleotides, and DNA is double strand. So, and this circles, each circle red, green, yellow, blue. This circles represent the circle is specific amino acids. We know that there are 20 different types of amino acids. So transfer RNA takes those amino acids, specific amino acids, and brings them in the ribosomes and join together amino acid chain, synthesized amino acid chain. So this one is synthesized amino acid chain, and this chain becomes protein or peptide. So another important thing is this mRNA. You can notice this tiny bar, little bar. Each of these bar is a single nucleotide. Nucleotide is basically adenine, guanine, cytosine, etc. We'll learn more about this structure of this mRNA or nucleotides in the nucleic acid chapter. So just know that the single bars are nucleotides, and each of these three nucleotides is called codon, that codes for a specific amino acid. So this every three every three nucleotides codes for one amino acid, a specific amino acid. So uh, nucleotides are basically adenine, guanine, cytosine, thymine. So based on the combination of those nucleotides, that specific combination encodes for a specific amino acid. So, and tRNA takes those specific amino acid and join them in the translation process. So this is, in a nutshell, is the translation process in a very simple figure. So the function of the ribosome is assemble amino acids to form specific proteins. We know that already, and this is called protein synthesis. The ribosomes read mRNA, messenger RNA. Messenger RNA actually carries the blueprints for the encoded or translated protein. So this mRNA and ribosomes, this Pac-Man reads this mRNA and those piece together amino acids. The ribosome piece together those amino acids with the help of transfer RNA. So this little guy, they will bring together amino acids. So basically the transfer means the transfer amino acids. So they are called transfer RNA, tRNA, and bring them to the right amino acid in sequence according to the specific codon. So you don't have to worry much now. Just step by step read through this. You can understand. These are very simple concepts. So moving to the next slide.